Today we discuss about the plant tissue culture. This is the first part of this lecture series. Mainly focus on history and basic terminology of plant tissue culture. These points are important for the TYBSc Botany as well as MSc student. So let's start. So first one is what is plant tissue culture. So plant tissue culture is a technique of growing plant cell, tissues, organs, seeds or other plant part in a sterile environment on a nutrient media. So mainly this slide we can understand the definition of plant tissue culture. So mainly in this definition some words are important for understanding this definition. So mainly we can select any part of the plant because plant having some properties. So we can select some cells, tissues, organs, seeds or any other part of the plant in the sterile environment. So mainly for the purpose of plant tissue culture we can select sterile environment because mainly growing plant or any tissues they have ability to some grow microorganisms. That's why we can select sterile environment for the avoiding of other microorganisms. And grow mainly cells are grow on the nutrient media. Okay, I think you easily understood what is meant by plant tissue culture. So next, the history of plant tissue culture. The idea of tutipotency of a plant cell was given by Haberland in 1902. So firstly, Haberland was discovered the tutipotency ability in the plant cell. What is meant by tutipotency ability? Tutipotency means the ability can produces whole plant body from the single cell. That's why Haberland known as the father of plant tissue culture. After the Haberland, White was the first time established successful root culture of tomato in the 1937. After next, Van Beek used coconut milk for growth and development of young Datura embryos in 1941. Skoog and Miller demonstrated the role of auxin and cytokinin on root and shoot formation in tomato plant in 1957. Generally, auxin and cytokinin both are the root or shoot hormones. Generally, they can grow the formation of root and shoot. It's firstly discovered or demonstrated by the Skoog and Miller. After Skoog and Miller, Murasinghe and Skoog introduced the medium for tomato culture in 1960. The first trial by the Murasinghe and Skoog on a nutrient medium for the purpose of tomato culture in 1962. After Murasinghe and Skoog, Guha and Maheshwari obtained for the first time haploid plants through anthroculture of Datura. In this slide, I have mentioned the year, worker and advancement in the plant tissue culture. So one by one we can discuss of these scientists work. In 1902, Haberland first attempt in vitro culture of plant cell. We already discussed. In 1904, Hanning was culture of embryonic tissue of crucifers. In 1922, Robbins was in vitro culturing of roots. In 1925, Liebig was culturing zygotic embryo in linum plant. In 1934, Voigt was culturing the root of tomato plant. In 1939, Guetharbet, Fight, and Knopfert successfully establishment of indefinite callus. In 1941, Brahm was culture of crown gall tissues. In 1945, Lou was culturing from stem teeth. In 1957, Stug and Miller discovered the auxin to cytokinin ratio regulate the organ formation. That means root formation as well as shoot formation. In 1960, Bergam was development of planting technique for isolation of single cell. In 1970, Power successful protoplast fusion. In 1970, Maheshwari and Guha was successfully culturing anther culture. In 1971, Tagbe was plant regenerate from the protoplast. In 1974, Reinhardt was the preparing biotransformation in plant tissue culture. In 1978, Matures was produces of somatic hybrid tomato plant. So these are the some workers or scientists work on the plant tissue culture. So some terminologies of related to the plant tissue culture. So first one is the adventitious. Adventitious means development from unusual point of origin such as shoot or root tissues from callus or embryos from sources other than zygote. Agar. Agar is a polysaccharide powder derived from the algae, mainly for gelidium, used to get a medium. Algae is the generally used a concentration of 6 to 12 gram per liter. Agar is the mainly solidifying agent in the medium. Next, aseptic technique means procedure used to prevent the introduction of fungi, bacteria, viruses, mycoplasma or other organisms into culture. Autoclave. Autoclave is the machine capable of sterilizing wet or dry item with steam under the pressure. Pressure cookers are a type of autoclave. Then callus. Callus is the unorganized proliferate mass of differentiated plant cell, a wound response. Mainly, it is an unorganized mass of the cell. We cannot distinguish mainly a root cell as well as shoot cell. So, it is an unorganized mass of cell. Then, chemically defined medium a nutritive solution for culturing cell in which each component is specifiable and ideally of known chemical structure. Clone. Plants produce asexually from a single source of plant. Clonal propagation. Asexual reproduction of clones that are considered to be genetically uniform and originated from a single individual or explant and will support the continued cell division of mature cell, leading to the formation of callus. Coconut milk. The liquid endosperm of coconut contains the cytokinin zeatin. Generally, it is a liquid endosperm which is present within the coconut. Then contamination. Contamination means being infested with unwanted microorganisms such as bacteria or fungi. Culture. Culture means plant growing in vitro condition. In vitro means lab condition. Detergent is the increasing of efficiency of sterilization. Differentiated cells that maintain in culture all or much of the specialized structure and function typical of the cell type in vivo. In vivo means outside the lab condition or inside the body. What is vivo? So modification of new cells to form tissues and organs with a specific function. Explant. Explant is the tissue taken from its original site and transferred to an artificial media for growth or maintenance. Mainly, we can know the explant is the plant material for the selection of plant tissue culture. Horizontal laminar airflow unit, an enclosed work area that has sterile air moving across it. The air moves with uniform velocity along parallel flow line. Room air is pulled into the unit and forced through the HEPA. HEPA is the high energy particulate air filter, which remove particles 0.3 μm and large. So generally, horizontal laminar airflow unit is the enclosed area for working inoculation. Inoculation means the transfer of explant into the culture media. Next one, hormones. Growth regulators, generally synthetic in occurrence that strongly affect growth, that is cytokinin, auxin, gibberellic acid, ethylene. So is the plant growth regulator known as the hormones. Internode, the space between two nodes on a stem. Media means a flora is a medium. Media is the culture media. They having different types of nutrient like a micronutrient, micronutrient, carbon source, vitamins, some hormones. So medium is the nutritive solution. It's the solid or liquid for culturing cell. Micropropagation in vitro clonal propagation of plant from shoot tip or nodal explant, usually with a accelerated proliferation of shoot during subculture. Node, a part of the plant stem from which a leaf, shoot or flower originate. Pathogen is the a disease causing organism. So pathogenic means capable to causing a disease. Petiole is a leaf stalk, the portion of the plant that attach the leaf blade into the node of the stem. Plant tissue culture, the growth or maintenance of plant cell, tissues, organs, or whole plant in vitro in lamp condition for aseptic condition. Regeneration. In plant culture, a morphogenetic response to a stimulus that result in the product of organ, embryos, or whole plants. Somaclonal variation. Phenotypic variation, either genetic or epigenetic in origin, displayed among somaclones. Somaclones, plant derived from any form of the cell culture informing the use of somatic plant cell. This culturing the plant cell mainly for plant having two types of cells, some of the somatic cell or gammatic cell. So somatic cell is the body cell. Formation of or dividing of the body cell or somatic cell called as a somaclone. Sterile. First one is the without life. Sterile means without life. Second one is the inability to produce organism to functional gamete. Means those organisms do not produce functional gamete for the reproduction. What is a sterile organism? A culture that is free for viable microorganisms is also called as a sterile environment. So sterile environment means the free from microorganisms. Okay. So let's sterile technique. A practice of working with culture in an environment free from microorganisms. Then culture. See passage with plant culture, that is the process by which the tissue or explant is first subdivided, then transferred into flesh culture media. Means generally the firstly we can inoculate the explant, and they after day by day the formation of various tissues, various cells within the nutrient media. But for the purpose of passage or space, we can curve the callus and again culture with different bottles for the space, availability of space. That's called as a subculture. The tissue culture, the maintenance or growth of tissue in vitro in a way that may allow differentiation and preservation of their function. Totipotency is a characteristics in which the potential for forming all the cell types in the adult organisms are retained. Mainly, in simple way, totipotency is the ability of cell which can grow or which can create, which can divide whole plant body from a single cell. It is the ability of plant cell. But in animal, only 4 cell stage of embryo which can have the totipotency ability. Stem cell having totipotency ability. But in plant cell, every cell having totipotency ability. Undifferentiated means with plant cell existing in a state of cell development characterized by isogametic isodiametric cell shape, very little or no vacuole, a large nucleus, and exemplified by cell comprising a apical meristem or umbel. These are the some terminology regarding the plant tissue culture. But why plant tissue culture are important? So the production of clone of plant that produce particularly good flower, fruits, or have other desirable traits. Next, to quickly produce mature plants. The production of multiplies of a plant in the absence of seed or necessary pollinator to produce seeds. Generally, some endangered plant species which can produce a multiply plant for the purpose of growing growing and increasing the number, the regeneration of whole plant from plant cell that have been genetically modified. So how tissue culture occurs? Adult plant cells have pluripotent, meaning they have the ability to give rise a fully differentiated plant. Because of these, it is possible to collect cells from a mature plant and use those cells to produce clone of that plant. So mainly here the root of carrot plant, and from the root of carrot plant we can isolate some cells for the purpose of tissue culture. So these cells known as the explant, So some cell isolate from the root of carrot plant. So this carrot plant known as the mother or parent plant. So mother plant explant collect from the mother plant. So mother plant having some character. Which character we considered in the mother plant or parent plant? Firstly, it is disease resistant or disease free plant. It is a genetically pure plant. It is high yielding capacity. In that plant having high yielding capacity as well as high nutritive value. So explant sterilization, after the sterilization of explant is transferred to the nutrient medium. So explant transferred to this nutrient medium under the sterile condition called as inoculation. So after the inoculation, these slant or bottles, tissue culture bottle, placed in the maintained condition or aseptic condition called as incubation. After the incubation, some days or some duration, these explant or these cell multiply in the aseptic condition on the nutrient media. So single cell divide into two cell is converted into 4 cell, and 4 cell into the undifferentiated mass of the cell called as the callus. So after the callus, we can done the subculturing. After the subculturing, we can, for the purpose of subculturing, we can use hormonal ratio in the nutrient media. So some are the root hormones, some are the a shoot hormone, and we can use this hormone and form the young plant. Young plant formation. After the young plant formation, this plant capable to grow on natural condition, so called as the hardening. Called as a hardening. And after the hardening, we can grow of this plant in the natural condition and known as the tissue culture plant. Okay, so this is the general procedure for the plant tissue culture. Detailed procedure of plant tissue culture we will discuss in the upcoming lectures. Okay. So basic requirement and techniques of plant tissue culture. The main requirement of plant tissue culture are the laboratory organization, culture media, and aseptic condition. These three requirements are needful for the plant tissue culture. So first one is the laboratory organization. In a standard tissue culture lab, there must be a few basic facilities like a media room for preparation, sterilization, and storage of culture media, facilities for washing of labwares, explant, etc., space for storage of labwares, culture room or incubator where condition of temperature, humidity, and light, etc., can be maintained, observation and data collection area. So these are the few basic facilities are must in the laboratory organization. Then culture media. The formulation or the medium on which the explant is cultured is called culture medium. It is composed of various nutrients required for proper culturing. Different type of plant and organs need different composition of culture media. A number of media have been devised for specific tissues and organs. Mainly some important of them, some important media like MS media. Long form of MS is the Mula and Scoop. Both are the scientists who discovered the medium, first medium known as the MS media, and MS media is the very popular maximum plant to grow in this media. Then LS media, Lensmeyer and Scoop medium. Then B5 media, Gamrose media is mainly for the banana, white media, so etc. These are the few media. Important constituent of culture mediums are some organic supplement like vitamins like thiamine, B1, pyridoxine, B6, nicotinic acid, B3, etc. Antibiotics like streptomycin, kanamycin, amino acid like arginine, asparagine. Some inorganic nutrients, micronutrients as iron, manganese, zinc, molybdenum, copper, boron. Macronutrient include 6 major elements as nitrogen, sulfur, phosphorus, potassium, calcium, magnesium. Carbon and energy source. Most preferred carbon source is the sucrose. Other including lactose, maltose, galactose, raffinose, cellulose. Some growth hormones are there like auxin, mainly for inducing cell division, cytokinin, mainly for modifying apical dominance at shoot differentiation, Abscisic acid like ABA used occasionally. Gibberellic acid is also called as a gibberellins, used occasionally mainly for the internodal growth. Gelling agent, these are added to media to make them semi-solid or solid like agar, gelatin, alginate, etc. Are common solidifying or gelling agent. Other organic extract, sometimes culture media are supplemented with some organic extract also like coconut milk, organic juice, tomato juice, potato extract, etc. Next one is the aseptic condition. Maintenance of aseptic condition is the most critical and difficult aspect in vitro culturing experiment. Aseptic condition mean the condition free from any type of microorganism so as to prevent the loss of experiment by contamination. For this sterilization, that is the complete removal or killing of microbe, is done. The most common contamination is in culture are fungi and bacteria. Some factors affecting plant tissue culture. First one is the growth media. Minerals, growth factor, carbon source, hormones, and their concentration may also affect plant tissue culture. Some environmental factors like light, temperature, photoperiod, media is also affecting for the plant tissue culture. Explant source, usually the younger, less differentiated, the explant is better for tissue culture. If you select older explant, then it's also affecting factor for the plant tissue culture. So this is our first lecture of the plant tissue culture. So these are the references used for preparing this presentation. Thank you.